Hello students, in this video we're going to go ahead and get into the last set of notes for Unit 4, the 4.2 notes, which is all about the cell cycle. And so in these notes in topics 1 and 2, we're going to be talking about what the cell cycle is and the different phases that make up the cell cycle. And then in topic 2, we're going to talk about how ultimately the cell cycle is controlled and regulated inside of cells. But in this video, we're going to go through topic 1. In topic 1, we're just going to go through what happens during the cell cycle. So the cell cycle is basically you can think of it as the life of a cell, starting from when that cell first formed and these different stages of life that cell is going to go through before it ultimately divides into two new cells, which is what happens to a lot of cells towards the end of their life. They then divide and form new cells. And so the cycle of a cell, or the life of a cell, consists of two main phases. There's interphase, which is the majority of a cell's life, and then there's the mitotic phase, which is at the very end. Interphase is when the cell is going to be growing and doing normal cell functioning things that depend on the type of cell. So whatever that cell is supposed to be doing, it's doing that during interphase, and it's growing and becoming a larger cell. And that's also when during interphase, at some point, the cell will actually take its chromosomes and make a copy of its chromosomes in preparation for the mitotic phase. In the mitotic phase, that's when the cell is going to actually then divide into two identical daughter cells. So we're going to go through a lot of the different steps involved in all of this, starting with interphase. So like I said, interphase is the majority of a cell's life. It's about, on average, 90% of a cell's life is spent in interphase for a typical cell. Like I said, this is when a cell is performing its usual function. So if it's a muscle cell, it's doing muscle cell things, or a skin cell doing skin cell things, and so on. Within interphase, we can actually divide it into three subphases. There's a phase called G1, then S, and then G2. And the G1 phase, the G stands for gap. When scientists first came up with these phases, it looked like during the G phases, like G1 and G2, there wasn't a lot going on. We've since learned, though, that a lot does go on in a cell during these phases. But that's why the G comes from. It comes from calling it like the gap phase, the first gap phase. But this is when this is the first phase of the cell cycle, and this is when the cell is growing in size. And during this phase, a lot of cells start duplicating their organelles. And so they take their mitochondria and make copies of them. The endoplasmic reticulum and the Golgi, they start making copies of these organelles in preparation for one day dividing into two cells. Because if you're going to divide into two cells, you're going to need an extra set of everything for those two new cells to form. And then there's the S phase. And the S phase, this is actually probably the most important phase during interphase. The S stands for synthesis, and this is actually when the cells are going to synthesize a new set of DNA. So they're going to take all the DNA that's in the cell, which is the blueprint of the instructions for the cell, and they're going to make a copy of all of the DNA. And inside the cell, the DNA exists as chromosomes, which are different huge pieces of DNA inside the cell that have all these different genes and instructions on them. And basically in that cell, it's going to make a copy of all that DNA. It's going to replicate it, which is going to cause the chromosomes to duplicate. So all the chromosomes that are in the cell, they're now going to be duplicated. We're going to make a second copy of all of those chromosomes containing all of that DNA inside the cell. Again, for the purpose of one day dividing, like you're getting ready to divide into two daughter cells, and each of those daughter cells are going to need a copy of all those instructions that are in the DNA. Those two copies of the chromosomes will be held together as sister chromatids, which you guys will see in a second. And then after the S phase, the cell will then enter the G2 phase, or the second gap phase, and this is when the cell is going to continue to grow and makes final preparations for the mitotic phase, which is when the cell will actually divide. So at the end of interphase, if the cell has gone through G1, S, and G2, here's basically where a cell's at before it starts the mitotic phase. At the end of G2, so like right here, before it's starting the mitotic phase, here's what the cell would here's some important things that are going on in the cell at this time. First of all, there's the nucleus and the nuclear envelope, which is this membrane that surrounds the nucleus. It's still here, and inside that nucleus, you have all of the chromosomes, all of the DNA, but all that DNA has been duplicated And so you're basically going to have twice as much DNA in here now, because during the S phase, all of this DNA was copied. But it exists as this huge mess of chromatin in here, so it's all uncondensed and just all tangled up in here in the nucleus. And you'll see in a second what's going to happen is the chromosomes will start condensing. But right now it's just all loosey-goosey here in the nucleus with these chromosomes. And then you have these special organelles called centrosomes. We talked about organelles inside of cells in Unit 2, but we didn't really talk about these guys. These are called centrosomes. A centrosome is the organelle that's actually going to be organizing these microtubules. There's these special proteins that we're going to see in a second that are actually organized here in the centrosomes, and they're basically going to orchestrate all of the events that are about to take place during the mitotic phase. So basically, these centrosomes are really important for the mitotic phase to take place, because it's going to involve these microtubules that are organized in something called a mitotic spindle, which you'll see in a second, all because of these guys. But anyway, during interphase, the centrosome was duplicated, so there's 2 centrosomes. Each of those centrosomes contain a pair of centrioles. So these little cylinder-looking things, those are called centrioles. And in one centrosome, like this right here, this is one centrosome, and it has a pair of centrioles. And here's another centrosome. It's been duplicated, has another pair of centrioles. So now there's 2 centrosomes inside the cell. At the end of interphase, at the end of G2, and you have twice as much, all your DNA has been replicated, it's here inside the nucleus, and this is where we're at so far. So then, now we're ready for the mitotic phase, and the mitotic phase, which is going to take place after interphase, this is when the cell is actually going to start dividing and doing all the things necessary for the cell to ensure that the 2 daughter cells that get produced are produced correctly. And so in the mitotic phase, there's 2 parts to the mitotic phase. There's mitosis, and then there's something called cytokinesis. During mitosis, what we're going to do is take the nucleus and the DNA that's inside of the nucleus that's already been replicated, and we're going to divide it into 2 new nuclei, plural. We're going to take the nucleus and divide the nucleus between these 2 future cells that are going to form. And then in cytokinesis, that's at the very end, and that's when we're actually going to split the cytoplasm between the 2 cells and form 2 distinct separate daughter cells during cytokinesis. And so we'll look at these more closely in a second. In mitosis, there's going to be 5 subphases involved. And so if you look here, there's prophase and then something called prometaphase, which honestly for this class, we kind of talk about prophase and prometaphase together, kind of as like one phase. And then there's metaphase, anaphase, and then telophase. So there's 5 subphases here, but really we're going to mostly just pretend that prometaphase is the same as prophase. We're going to talk about it as one phase. And so the acronym that you can remember these phases in is PMAT, if you haven't heard that before. But you have prophase, and then metaphase, the anaphase, and telophase. That's the order of phases that's going to happen during mitosis. And all of that's going to end with cytokinesis at the very end, when this cell actually splits into 2 different cells. So now what we're going to do is look at each of these phases and see what's taking place in these phases. The first one is prophase and prometaphase. And this will be, prophase and prometaphase is the hardest phase because there's a lot going on in prophase and prometaphase, and I'll show you guys in a second. But then after that, it's really easy. So during prophase, at the very beginning of mitosis, after completing interphase, one thing that's going to happen is the chromosomes are going to condense. And so all of that DNA that was loosey-goosey inside the nucleus, all that DNA is now going to actually condense into these very tight, these very tightly compacted, organized structures that form what these chromosomes, like typically when you guys think of a chromosome, you probably think of something like this. And this is all of that DNA, that giant piece of DNA, all condensed together in this structure. So this is that's going to happen during prophase. Is all those chromosomes are going to condense into these nice little tight packaged things right here. And what you're going to see is this structure here, where it actually is made of 2 pieces. We call them sister chromatids. These are the 2 copies of the chromosome. So in one of these sister chromatids, like this piece right here, this is all of the DNA that contains all of the genes that are located in this chromosome. And then during S phase, if you guys remember, during S phase of interphase, we actually made a copy of this. And so that's where that's what this structure is over here. This is the other structure, the copy of the chromosome. So these are the duplicated pieces of the chromosome that are still held together as one piece. And so we call these sister chromatids. So sister chromatids are exact copies of each other of that chromosome. And that's what you're going to see in prophase. Is you're going to see when these chromosomes condense, you're going to see a structure like this made up of two sister chromatids. Those sister chromatids are held together in the middle in a central region called the centromere. And then at the centromere, you're going to have these two protein complexes, one on each sister chromatid, called kinetochores. And those will be important in a second, you'll see. But this is what a condensed duplicated chromosome looks like. So when the chromosome condenses up into this tight little package, and it's already been duplicated during S phase, it's going to look like this, containing two sister chromatids attached together, which are two copies of that chromosome. And so during prophase and metaphase, like I said, you're going to see all of that DNA condense into these nice little packages, these condensed chromosomes here. And it depends on the organism, but every organism has a different number of chromosomes. So in this picture, it looks like there's 6 chromosomes here, 1, 2, 3, 4, 5, 6, that have all been duplicated. You can see the two sister chromatids on each of these, and they're all condensing now during prophase and prometaphase. So instead of looking like this huge mess right here, it now looks like this. They're all condensing up into these nice little condensed, tightly packaged structures. So that's one important thing that's happening during prophase and prometaphase. If this was a human cell, you'd actually see 46 chromosomes, so 46 of these guys. But in this example, we're just going to look at 6 because 46 would be a lot to look at. And so another thing that happens during prophase and prometaphase is the nucleus, the membrane of the nucleus, which we call the nuclear envelope, is actually going to be taken apart. It's going to be degraded. So inside the cell, they're actually going to take down and take apart the nuclear envelope. And so the nucleus is basically going to go away because the nuclear envelope is going to be degraded. And so this will be the only time in a cell's life where the DNA is not protected by the nucleus. But luckily, since it condensed up into these nice little packages, by it condensing up into these condensed structures here, it helps to protect the DNA while it's not protected by the nucleus. But anyway, the nucleus is going away. And then the last thing you're going to see is these centrosomes moving to opposite sides of the cell. So these centrosomes, which we have 2 of them now after interphase, they're going to start moving to opposite poles of the cell and start forming what's called the mitotic spindle. The mitotic spindle are these microtubules, which are these protein filaments that are being built by these centrosomes, and they're starting to grow, these creepy little fingers, and push each other away to opposite sides of the cell. And then these guys, these mitotic spindles, which are made of these microtubule proteins, they're going to actually start connecting to some of these chromosomes, and they connect to the chromosomes on the kinetochore. So this piece right here, this protein structure on the chromosome, that's where the microtubules are going to start attaching to these chromosomes. And so that's what's happening in prophase and prometaphase, we can just call it prophase, which is a lot. I told you it was going to be a lot, but just to recap real quick, this is when the chromosomes are going to condense into these tight little nicely packaged structures. The nuclear envelope is going to be degraded. We're going to get rid of that. And then the centrosomes are going to move to opposite sides of the cell, and the mitotic spindle is going to start to grow and start to attach to some of these chromosomes, which is going to start maneuvering these guys to where they need to go. And so after that, it actually becomes very easy. So after we do all of this, we then enter metaphase. And metaphase, those chromosomes are now going to be lined up in the middle of the cell. So those condensed chromosomes, which are made up of 2 sister chromatids, which are exact copies of each other, they're going to line up in the middle of the cell, just like this. And this is when all of the microtubules will be attached, this is when all of the chromosomes will be attached to microtubules. So these microtubules are going to attach to the kinetochore on each sister chromatid on this side and on this side. It's going to attach on this side and on this side. It's going to attach on this side and on this side to all of these chromosomes, and it's going to line them up in the middle. That's metaphase. And then they're going to be pulled apart. And so what's going to happen now is these microtubules that are attached to the kinetochores, they're going to actually shorten. So these microtubules are going to start breaking down and getting shorter and shorter and shorter. And as they're getting shorter and shorter and shorter, they're going to pull apart these duplicated chromosomes. So these sister chromatids that were held together, we're now going to pull them apart. Which again, remember, these are exact copies of each other that were held together at the centromere, but now we're officially going to separate them into 2 separate chromosomes. And that's what's going to happen to all of these chromosomes during anaphase. Their sister chromatids are going to be pulled apart towards opposite sides of the cell by these microtubules. And then lastly, you have telophase. During telophase, that's the last phase of mitosis. That's when, so once these chromosomes, these sister chromatids get pulled to opposite sides of the cell, they're going to start to uncondense back into the chromatin, the loose, untightly compacted DNA, the loosey-goosey DNA in the nucleus. They're going to start to uncondense. You guys can see that happening in this picture. And that's when we're going to start building a new nucleus around each set of chromosomes that have been separated now. And so now that they've been pulled to opposite sides of the cell, we need to now rebuild the nucleus around them. The nuclear envelope is going to be rebuilt, and you're going to end up with 2 new nuclear envelopes now surrounding these 2 sets of DNA that have been separated in the cell. And then lastly, you end with cytokinesis. So after mitosis and you finish prophase, metaphase, anaphase, telophase, you then have cytokinesis. In cytokinesis, that's when the cytoplasm is actually going to split between these cells, and you're going to have 2 distinct cells now exist, 2 daughter cells. And that's going to look a little bit different in animal cells versus plant cells. In animal cells, there's this pinching in of the cytoplasm. The cell membrane actually pinches inward, and then it will eventually cut into 2 different daughter cells. That's what happens in animal cells. But in plant cells, it looks a little different, because in plant cells, if you guys remember, there's a cell wall, and that cell wall is very rigid and strong and sturdy. And so the cell's not going to pinch inward. The cell membrane is not going to pinch inward. Instead, we're going to actually build a cell membrane and a cell wall in between the 2 daughter cells. So as those 2 nuclei get rebuilt and those chromosomes were separated, you're going to then see this cell wall get built between these 2 daughter cells to fully divide the cytoplasm of these 2 daughter cells. So now they're 2 distinct cells. And so that formation of a cell wall being built between these cells is called a cell plate. So you'll see a cell plate forming between plant cells during cytokinesis, versus in animal cells, you see what's called a cleavage furrow being formed before the cytoplasm gets separated into 2 distinct cells, just like what's happening here. So it's a little bit different in animal cells and plant cells, but you end up with the same thing. You now have 2 distinct daughter cells at the end. The way you guys can kind of remember PMAT, so prophase and prometaphase, think prophase, P for prophase, which means P for prepared. Like, this is when it's going to prepare a lot of different things. A lot of things happen during prophase, which we just talked about. And then metaphase, M for metaphase, but M for middle. That's when they line up in the middle of the cell. So think middle, metaphase, metaphase, middle. And then anaphase, think A for anaphase, A for apart. So that's when the sister chromatids get pulled apart for anaphase. And then telophase, that's when you end up with 2, T for telophase, T for 2 nuclei. So 2 new nuclear envelopes are going to form, forming 2 new nuclei for each of these daughter cells that are forming. And then you have cytokinesis, which literally means cutting the cell into 2 pieces. So those are the different phases. You guys do need to know those phases and what's going on in those phases. Hopefully, after you rehearse it a couple times, you'll realize it's not a whole lot going on. Especially if you can just get past prophase and prometaphase, there's a lot going on in those phases. And again, for this class, you guys can think of them as one phase. Just call it prophase. But there is a lot going on during that. And so here's a picture that shows you, like, in a real cell, what a cleavage furrow looks like in an animal cell when it's doing cytokinesis, forming 2 daughter cells, versus in a plant cell, what that looks like when the cell plate is forming. Again, doing cytokinesis, forming 2 distinct daughter cells. And then there's a few more things left in these notes for topic 1. Just an overview of the mitotic phase. So what happened during mitosis and cytokinesis is we started with an original cell, and during interphase, those chromosomes were duplicated. If you remember, during S phase of interphase, we're going to copy all of this DNA and duplicate the chromosomes, but they weren't all condensed like this. This picture is showing them condensed, but in reality, they weren't condensed. They were all loosey-goosey in the nucleus. But basically, we duplicated all of them, and now we have these sister chromatids that make up each of these chromosomes. And they're going to condense during prophase, and the nucleus is going away. And basically, those line up in the middle, and we pull apart those sister chromatids into separate chromosomes now that exist in each of these daughter cells. So basically, you end mitosis with two identical daughter cells. These daughter cells are identical genetically to each other. They have the exact same DNA information because they both have copies of the exact same chromosomes. And they're identical to the cell that we started this whole thing in. And the whole thing before we started interphase at the very beginning of the cell cycle, these cells are identical genetically to the original cell. And so that's very important. And then it's also important to point out here that in mitosis, you're starting with what's called a diploid cell, and you're forming two new diploid cells. And so I just want to explain what a diploid cell is, because we're going to talk about this a lot over the next couple units in this class. A diploid cell is a cell that contains two sets of chromosomes. So it has a set of chromosome from your mom and a set of chromosomes from your dad. So in this example, they kind of color-coded it. You can see there's 6 chromosomes in this cell, but it's actually 2 sets of 3. So there's 3 chromosomes that this cell got from one parent and 3 chromosomes that this cell got from another parent. And so we call that a diploid cell because it has 2 complete sets of chromosomes, one from each parent. And then you duplicate all of them and separate them into 2 daughter cells. And these daughter cells still both have 2 sets of chromosomes, 2 sets of 3 in this case, one from mom, one from dad, one from mom, one from dad. But they both got a copy of each of those chromosomes. And so in your guys' body, all of your somatic cells are diploid. So in eukaryotic organisms, multicellular organisms, somatic cells are diploid. And by somatic cell, somatic means body cells. So these are all the cells in your body except your reproductive cells, which are your sperm and your egg. So all the cells in your guys' body are diploid. And that's true for all multicellular organisms. They're diploid cells, except for the reproductive cells, like your sperm cells and your egg cells, they're not diploid. Your sperm and your egg actually only have 1 set of chromosomes, not 2 sets of chromosomes, which we'll talk about that in the next unit. And so mitosis is basically going to involve somatic cells. It's always going to be body cells doing mitosis, forming new body cells, whether it's muscle cells forming more muscle cells, or skin cells forming more skin cells, or stomach cells forming more stomach cells. But those are always going to involve diploid cells, making more diploid cells, which are somatic cells, body cells. And so every species has a different diploid number. So for this fake example, the diploid number is 6. So in this cell that has 2 sets of chromosomes, there's 6 chromosomes total. That's the diploid number. And that's different for each species. So for humans, for example, our diploid number is 46. So if I took one of your body cells, one of your skin cells, one of your muscle cells, one of your nerve cells, one of your stomach cells, any cell I take from your body, there's going to be 46 chromosomes in it. That's our diploid number, which is basically 2 sets of 23. You got 23 chromosomes from mom, 23 chromosomes from dad. So 46 chromosomes total. And then very lastly on here, well, actually there's 2 more slides, sorry. Is in bacteria technically don't do mitosis because they don't have, bacteria only have 1 chromosome, and it's circular, 1 large circular chromosome. They don't have multiple chromosomes. They also don't have a nucleus. They also don't have centrosomes forming these mitotic spindles. And so it looks a lot different in bacteria, but the bacteria cells do cell division. They do divide. In bacteria cells, though, there's a process called binary fission, which is the process by which a bacteria cell replicates itself and forms 2 daughter cells. But we don't call it mitosis, but it's very similar to mitosis, but a little bit different. But basically, you're doing the same thing. You're taking the chromosome. So there's 1 chromosome inside the bacteria, and it's going to make a copy of that chromosome. And then those 2 copies of the chromosome are going to go to be pulled to opposite sides of the cell. And then the cell is going to split into two cells, like through cytokinesis. And then you end up with 2 daughter cells. So a lot like mitosis, but technically not mitosis. In this case, though, this is how bacteria actually reproduce. Like this one cell is the whole organism. Like the whole organism is this one cell. And then now when it divides through binary fission, you now have two new organisms. Like this is the offspring. Like these are two new bacteria cells, two complete organisms now, which is not what's happening in mitosis. In mitosis, we're just taking cells that exist in our bodies and making more of them, but that's not us reproducing. We didn't make more copies of ourselves, right? Like we didn't make new organisms. We just made new cells that are part of our organism, our one body that we currently have. And then the last slide on here is just the importance of cell division and why we need it, which you guys definitely need to know. So why do cells divide through mitosis? And so the ability for organisms to reproduce and make more cells is a characteristic of life, basically. It distinguishes living things from non-living things. Like non-living things can't just replicate themselves, whereas living things, they're made of cells that can replicate themselves and make more cells. And unicellular organisms, like I just said, when the cell divides, that's how they reproduce. So they're literally making more offspring. Like they're creating more organisms by doing that. Whereas in multicellular organisms, we rely on cell division not to reproduce. We're not doing mitosis to make offspring, but instead we're making more cells, which is important for these reasons. It allows us to develop. Over the course of our life, our bodies are changing. So you guys started off as a fertilized egg, and from that fertilized egg, it divided and divided and divided and divided, and you start developing different body tissues and different organs, and even throughout your life, going from a baby to a child to a teenager to an adult to an old person. There's all these stages of development that are taking place that rely on cells constantly dividing and making new cells to allow that development to occur. It also allows organisms to grow. So again, for you guys to go from a little baby to a fully grown adult, to become bigger, you literally are going to be made of more cells. And so you need more cells to be a bigger organism to grow. So cell division is important for that. And it allows the cells that you currently have to be replaced when those cells are broken or damaged or worn out. So you guys constantly every day are replacing old cells. So some of your cells, they get tired, they stop working, they're not good at doing their jobs anymore. And you guys are constantly replacing them. That's true for your skin cells and your blood cells and your stomach cells and all sorts of cells in your guys' body. You're constantly replacing the old ones with new ones that work better. So that's why cell division is important. Anyway, that's it for this video. I will see you guys later.